Okay, we'll go ahead and start with the muscular system. Let's start again at the cellular level. Remember, at the beginning of class, we said that the system was based upon starting at a molecular level. Molecules get together, make a cell. Cells get together, make a organ system and an organ, and then the collection of organs make organ systems to carry out functions in the human body. So, as we look at the muscle system, we start at a cell, a myoblast. And basically a collection of myoblasts will come together and form a muscle structure, carry out a particular action. All of the muscles in unison make up the muscular system. As you can see, here's an individual myoblast. This illustration is out of your book. This is a structure, a tissue, a muscle fiber. And as you can see, the collection of muscle fibers together are wrapped in a bundle, and we'll talk about the different layers here of this bundle. And actually make a particular muscle. You're familiar with the striations that are in skeletal muscles being our voluntary muscles. The lack of these striations would indicate that this would be a smooth muscle. Types of muscle function: we have skeletal muscles, we have smooth muscles, we have cardiac muscles. You've already looked at those on the histological slides, so you know the basic structure of these muscles. As you remember, skeletal muscles have striations and are typically in parallel bundles, peripherally lying nuclei. Smooth muscles actually lack the striations. They also are in longitudinal bundles. We see those mostly in digestive organs. We see them in the vessels of blood vessels, the smooth muscles and blood vessels. And then cardiac muscle, cardiac tissue, basically is striated, but it's more of a branching network. And then it also has the intercalated disc that, again, you're already familiar with. Skeletal muscles, again, voluntary muscles, long, shaped in cylinders or tubes, and I'll show you some more illustrations of those. Composition, basically protein. The protein structures are arranged in striated pattern. And then these particular tissue types are attached to bone, which will help carry out a particular function. So again, you can see the relationship between the muscular system and the skeletal system in being able to provide us locomotion, be able to manipulate things in our environment by using those two systems together. And here's another illustration that again starts at the individual muscle fiber. The fiber bundles become together, and the fiber bundles are then bound together to make a particular muscle structure. And then here at the proximal or the distal end, you would see a tendon, which would basically then attach the muscle to bone. The other thing that you need to appreciate, and that will be a test question, are the bundling of these muscle fibers. As you can see, there are tissue structures that will actually surround groupings of bundles. There are actually tissue fibers, if you look at it more closely, that surround each of the individual fibers of muscle, and then there is a covering of tissue that surrounds the entire muscle, and I'll go into a little more detail about these coverings in just a minute. There is the smooth muscle. Differs a little bit from the skeletal muscles in that we don't see the striations. Again, we see these in the visceral walls of blood vessels and in the intestinal system. The integumentary system also has smooth muscles, again surrounding blood vessels, regulates the blood flow to the superficial dermis. Skin needs circulation too. Smooth muscles are the erector pili muscles that are around the hair, make the hair stand up on the end. You see smooth muscle in the cardiovascular system. Now, not to be confused with skeletal muscle, not to be confused with cardiac muscle, said that wrong, not to be confused with cardiac muscle, but there is smooth muscle in the cardiovascular system as well, especially in the blood vessels. Obviously, there'd be smooth muscle in those structures. And then in the respiratory system, the digestive system, the urinary system, and the reproductive system, all of these systems contain smooth muscle. Now, what are some key factors to look at when you look at these systems? Now, other than the respiratory system, which does have a voluntary component, all of the other systems are involuntary systems. So for smooth muscle, absence of striation, and also this is an involuntary type of system. Cardiovascular system, we don't have any control over that. The heart's going to beat whether we want it to or not. It'll stop whether we want it to or not as well. The respiratory system, again, that's not necessarily a involuntary system. We can tell ourselves to take a deep breath and to breathe out. That's primarily the skeletal muscles, though, that we're inducing to be able to do that. Remember, there are muscles between the ribs. There are muscles of the diaphragm that are striated, so we can have some voluntary control over it. But again, we don't have to think to breathe, so it is an involuntary system. Again, smooth muscle involved there. In the digestive system, urinary system, reproductive system, again, these systems carry out their function whether we want it to or not, and basically these are involuntary systems that are controlled by the autonomic nervous system. Again, we'll go more into autonomic nervous system next semester. Smooth muscle, this is the arrangement. Again, I didn't give you smooth muscle on the regular histological exam because a lot of times it's confused with dense regular connective tissue. But again, you see the longitudinal bundles here in cross-section. You see the nuclei. Again, if you look at it on the illustration, you'll see the smooth muscle cells. You can see some dense bodies. These don't show up very well when you look at it histologically, so they're not confused with striations as you would see in skeletal muscles. Now, smooth muscle does contract, and you can see how the dense bodies, these intermediate filaments, are involved in the contraction of that. They basically, when they contract, will contract the muscle tissue that underlies these structures and works in contraction. For cardiac muscle, we see that's found primarily in the heart. This is striated muscle as well. Again, the branching pattern of cardiac muscle is much different than what we see in regular skeletal muscle. The cardiac muscle also has what we refer to as intercalated discs. These are basically the tight junctions which hold cardiac muscle cells together. Now, the structure of some of the muscles, some of the structures that you'll need to be familiar with, you'll need to know these definitions. The belly of the muscle is the enlarged fleshy body of the muscle. The fascia of the muscle, this is a layer of tough connective tissue. As I was showing you earlier, some of the layering, the epimysium, the perimysium, endomysium, sometimes is referred to as the fascial planes or the fascial layers. But this basically connective tissue wraps around the muscle tissue, kind of holds it into position, holds it into place. Specifically, when we talk about the layering around the muscle fibers, there's the epimysium, which is the most outer layer of fascial tissue. There's the perimysium that surrounds the individual or the small muscle bundles, and then endomysium surrounds each individual muscle fiber. And so you can go back to the illustration before, and I actually may have another one coming up here, where you can look at each of those coverings, epimysium, perimysium, and endomysium, and you need to be familiar with those different coverings. And then the fascicles are basically the small muscle bundles. Oh yeah, we do have another picture coming up. It shows you the epimysium, perimysium, endomysium, all of which make up a functional unit referred to as the sarcoplasmic reticulum. Again, as you look at these cross-sectional pictures, this illustration out of your book, you can see each of the muscle fibers, again, surrounded by the epimysium, the endomysium, and the perimysium. Now, when we talk about the sarcoplasmic reticulum, we're talking about a myofibril or a contractile unit of the muscle, and we'll go into a little more detail about that later on in the lecture. Now, there are some abnormalities that can affect muscles. One of the abnormalities that I would like you to be familiar with is referred to as the muscular dystrophies. These are inherited diseases that produce progressive muscle weakness and deterioration. This is one of the most common abnormalities and actually one of the best understood conditions or illnesses that we deal with. We see that it appears in childhood. Many of you may be familiar with the telethons or the programs that usually come on television looking for contributions to be able to help individuals that are affected by this disease. Duchenne's muscular dystrophy is the most common type. It affects males mostly. The incident roughly 30,000 to 100,000 male births. Progressive muscle weakness is noted here. The child typically requires a wheelchair by the age of 12. Most individuals will die by the age of 20 due to respiratory paralysis and or cardiac problems. Now, skeletal muscles are primarily affected, but again, because you see it will affect cardiac problems, it also will affect the respiratory system. It can have an effect on smooth muscle and cardiac muscle as well. Some of the reasons individuals' skeletal muscles are affected are still under research, still under consideration to find out why the large skeletal muscles, the large groups of muscles, like what we see in the lower extremities, are affected first. Facial muscles tend to continue to function normally until the latter stages of the disease, which tells you that muscular dystrophy affects the large muscle groups, the large voluntary striated muscle groups much more prominently first, early in the disease, and then later develops into these other organ systems. Now, we'll move back into muscle structure itself. Another structure that you need to be familiar with is referred to as aponeurosis. Aponeurosis. This is collagen fiber of the endomysium and the perimysium as they taper off either at the distal end or the proximal end of the muscle. They will taper off, they will blend, and then typically will attach into a bony surface. There are a couple of key points on the body that have large aponeurosis, but it's basically a flat collagen fiber that's interwoven between the endomysium and the perimysium. They blend and then will attach in a bone. At each end of the muscle, the collagen fibers, the epimysium, perimysium, and endomysium come together and form this bundle of tissue or this tendon. The primary function of aponeurosis or tendon, they usually attach skeletal muscle to a bone. At the point where they contact the bone, these collagen fibers will extend into the bony matrix and provide a firm attachment area. The results are when the muscle contracts, it will pull the bone. And when it pulls the bone, it carries out particular function, whatever that muscle and that skeletal structure is supposed to carry out. And I'll show you a picture coming up of aponeurosis. Now, the sarcolemma, this is the cell membrane which covers the muscle fibers. There are also points of attachment where cell membranes will penetrate deep into the interior of the muscle fiber. That's referred to as the transverse tubules, sometimes referred to as T-tubules. And then there are myofibrils, indicative of muscle fibers. Myofibrils, these are long cylindrical structures made of a series of contractile units, and again the contractile unit are the sarcomeres, as I showed you a little bit earlier in the illustration. Two protein fibers that are key in muscle functioning are actin and myosin. So each contractile unit, the sarcomere, extends from what's referred to as Z-line to Z-line, and this will make more sense when I show you the illustration. But basically the contractile units form a unique system of contractile proteins. There are thick proteins which make up thick filaments, the thick myosin filaments. They sit between actin filaments. The actin filaments are the thin filaments that extend toward the center of the sarcomere. And they actually overlap. And because they overlap, as they contract, the muscles will contract. And it's basically this series of reactions that take place between the protein filaments that allow muscles to relax and to contract. And again, in the illustration, you can see that a lot more. If you look at the individual alignment of the actin filaments, this is the Z-line. These are the fiber elements that hold these muscle tissues together. And they basically give you what appears to be a Z-like pattern. The reason why you see that is because when contraction takes place, literally muscle fibers will overlap. When this aspect, when the thin protein filaments contract, let me go back. When the thin protein filaments contract, it actually will pull a series of muscle fibers in this direction, say toward the left, and then it will also pull a series of muscle fibers to the right. And thus, these two elements, these two muscle fibers will overlap. And that's what causes a muscle contraction to take place. So when you bend your arm up and your biceps muscle contracts, it does so because these actin and myosin filaments are overlapping and contracting based upon its arrangement around the Z-line. Now, there are other protein elements involved, as you can see in the illustration as well. And I'm actually making it a lot more simplistic than it really is. Your book goes into a lot more detail about it. But the basic concept that I want you to take away from here, these muscle fibers, these protein fibers, and these myofibrils, each of these lie, they're stacked upon each other, and when contraction takes place, they move. They basically are just overlapped, and that's how muscles contract. Now, cardiac muscle, as you remember, is not so longitudinal, and skeletal muscle, functioning of the skeletal system throughout our body, most of the voluntary muscles there are basically in parallel bundles. There are some little different arrangements, but basically they can easily overlap. The dynamics of the heart is a little bit different because you remember the heart, well, you don't remember, you hadn't had AMP2 yet, sorry. But the heart contracts at the top, contracts at the bottom, contracts at the top, contracts at the bottom. So that series of longitudinal muscle fibers wouldn't work too well in the heart because the heart functions a little bit different. It has to be coordinated, but the muscle fibers contract differently. There are chambers in the heart, so some of the muscles have different patterns. So it's not possible for the heart to have this long cylindrical structure like we do, say, in the muscle of the arm, the muscles of the forearm or the lower extremity. And so you see the muscles arrangements are a little bit different, but the same principle is still true. It's just the way the protein fibers and the muscle fibers are laid out in the cardiac muscle is a little bit different. This is basically, again, a muscle fiber. Again, skeletal muscle, peripherally lying nuclei, the complete surrounding of the muscle fibers, again, the endomysium, perimysium are located there. And again, you can see here the arrangement of each of these muscle fibers. The illustration does a good job of also pointing out to you where the Z line would be located at, where these muscle fibers will overlap and contract. It's basically a nice picture demonstrating the contractile unit of a muscle fiber. Now, I told you we'd come back to the sarcoplasmic reticulum. This is where these transverse tubules basically encircle the myofibrils, or the muscle fibers. The tubules are tightly bound to the membrane of the sarcoplasmic reticulum. This membrane complex is similar to the smooth endoplasmic reticulum that we talked about. We talked about in cellular anatomy. In skeletal muscle fibers, we see that the sarcoplasmic reticulum forms a tubular network around each individual myofibril, which will help in the muscle contraction. Now, the important part of all of this is, well, not just the important part, but another important part of this process is the communication between the muscle and the nervous system. Now, we already know there is a significant relationship between the skeletal system and the muscle system. The skeletal system cannot function without the muscles, and the muscles are basically jello without the skeletal system. So those two systems obviously have to work together, but then there has to be the nervous system that tells the muscle system what to do. So it's important that the nerves communicate with the muscles, and they will communicate with the muscles at a point referred to as the neuromuscular junction. So skeletal muscle fibers contract only under the control of the nervous system. Communication between the nervous system and the skeletal muscles occur at a specialized intracellular connection known as the neuromuscular junction. I know that's on the test. Neuromuscular junction. Now, what happens? There is an arrival of an action potential, a stimulus starting at the brain, sends a signal through a series of nerves, comes all the way down to your little pinky finger and tells your little pinky finger to move. So the stimulus comes from the brain, sending a signal, and when it gets to the point of the muscle, it will release what's referred to as a neurotransmitter. A neurotransmitter is a chemical substance that's usually at the very end of that nerve that's released, and it makes a chemical reaction take place between the nerve and the muscle. Opens up calcium gates that we'll talk about just a little bit, able to transmit that stimuli, that electrical stimuli to the muscle, causing a contraction to take place. So the stimulus for acetylcholine release is the arrival of an electrical impulse or an action potential at the synaptic terminal. Synaptic terminal is the very end of the nerve. The action potential is a sudden change in what causes a sudden change in what's referred to as a transmembrane potential. It's basically where the electrons are the negatively and positively charged ions, I should say, not electrons, negatively and positively charged ions will basically line up on the surfaces between the nerve and the muscle. And when this impulse comes through, it changes the electrical potential of how these cations and anions are lined up. When it changes the potential, we see specific gates open up. The gates will allow calcium to pass through. With the calcium comes the electrical impulses, and it causes this stimulus to be transmitted to the muscle. Now, I make this long speech about this reaction. It happens instantaneously, and these reactions take place consistently all the time in our body, 24 hours a day, 7 days a week, whether that's from voluntary muscles or involuntary muscles. Remember, even when we sleep, our system is working, so these stimuli and this system has to be working 24 hours a day to make our body function appropriately. This illustration is out of your book again, demonstrating to you the neuromuscular junction. This is the axon of a nerve extending and attaching at the neuromuscular junction. Neuromuscular junction is basically the attachment of the nerve to the tissue itself, to the muscle tissue itself. And it's at this point of attachment, where you'll see the release of acetylcholine. Your book goes through some steps in this process as well, and I'll go through those as well for you. Also, I'll just point out, as you can see, you remember mitochondria. Mitochondria are the little powerhouses of cells, supply energy. And in muscular contractions, muscular activity, we require mitochondria to be available, because mitochondria will supply the energy for these processes to take place. But this is a good illustration of the neuromuscular junction. Again, the nerve transmitting a signal. Again, the signal stimulates the release at the synaptic terminal, release of acetylcholine. Acetylcholine will then change the transmembrane potential, will release or open up calcium gates. Calcium will then pass out of the cell. Electrical impulses will pass with it, stimulate the muscle fiber for a contraction to take place. Again, this illustration out of your textbook, illustrating the neuromuscular junction. Go back and appreciate the neuromuscular junction. Sorry, get my arrow to work. The neuromuscular junction. This is the neuromuscular junction. We'll see the arrival of the action potential, which is the electrical stimuli that comes through, the release of acetylcholine, the neurotransmitter. Changes here, we'll see the neural membrane, the synaptic cleft. This is the point of contact between the nerve and the muscle itself. We'll see acetylcholine will bind at a portion referred to as the motor end plate. Again, that's the muscle side of the connection between the nerve and the muscle. And then we'll see the appearance of the action potential at the contractile unit, the sarcolemma. The action potential then spreads across the surface of the sarcolemma. That's when the acetylcholine is released. When we see acetylcholine released, it has to be used again. And acetylcholine then must be inactivated immediately. And there's another chemical substance that's produced that will inactivate acetylcholine, and that's referred to as acetylcholinesterase. Acetylcholinesterase, the ASE tells you it's an enzyme specific to inactivate acetylcholine. Don't want the muscles to go into tetany. And so when acetylcholine is released, acetylcholinesterase comes in. It doesn't really destroy acetylcholine, it just allows it to be reprocessed and reabsorbed, and can be used again later. And then the muscles go back to their original or their initial state. And this process goes on a continuous basis, again, 24 hours a day, 7 days a week. A little bit more about acetylcholine. The cytoplasm at the synaptic terminal, again, this is the point where the nerve will make contact with the muscle. The synaptic terminal contains mitochondria. We need mitochondria for energy, and vesicles that's filled with molecules of acetylcholine. Acetylcholine is a neurotransmitter, chemical released by the neuron to change the permeability or other properties of another cell membrane. This is that membrane potential to allow the passages of certain anions and cations. Cations specifically would be calcium. The release of acetylcholine from the synaptic terminal can alter the permeability of the sarcolemma, just the covering of this muscle contractile unit, and then will trigger the contraction of muscle fibers. Acetylcholinesterase again, acetylcholine dissociates or leaves the binding site and is immediately destroyed used loosely, destroyed by acetylcholinesterase, which is basically an enzyme that's found at the neuromuscular junction near the muscle membrane. The free binding sites are then ready for additional acetylcholine when the nerve is stimulated again. So this allows the muscles to completely to recycle and to continue to carry out their process. There's this series of reactions that continues to happen throughout the activity of muscle. Now, the synaptic cleft, again, is that little space between where we see the nerve will end and it will join and will attach to the muscle. Narrow space separates the synaptic terminal of the neuron from opposing sarcolemma surface, which is on the muscle. The surface contains a membrane receptor that will bind acetylcholine. At this point of binding, that's referred to as the motor end plate. At the synaptic cleft, the sarcolemma also contains molecules of enzymes, acetylcholinesterase, and again, their role is to destroy the acetylcholine so the recycling can take place, so more acetylcholine can come into the area. So even before the action potential has spread completely across the sarcolemma, or the contractile unit of the muscle, acetylcholine has been broken down by acetylcholinesterase. So this happens instantaneous, almost immediately upon the release of acetylcholine. We'll see acetylcholinesterase come in and break it down. Some of the breakdown products will be reabsorbed back into the synaptic terminal, used to resynthesize acetylcholine for subsequent release. So yes, it's destroyed, but most of the products are retained, can be reabsorbed and used later. The sequence of events can now be repeated should another action potential arrive at the synaptic terminal. And this is just another illustration, again out of your book, that shows those series of steps. Shows you the nerve, the motor end plate, the synaptic terminal, the release of acetylcholine, the binding that takes place, the release of calcium, the primary cation that's involved in this reaction, allows the action potential to come through, reach the point of the muscles at the T-tubule, stimulates the reaction to take place, the contraction of actin and myosin, which allows the muscles to overlap, and then the removal of acetylcholine by acetylcholinesterase, and then we'll see another action potential is now the muscle returns back to its normal state, and then another action potential can come through and the process starts over again. Those steps are listed for you in your textbook. I won't read those out to you. Now, some other muscular disorders that you should be familiar with. Now, these muscular disorders occur at the point of the neuromuscular junction. Diseases can interfere with muscles, can interfere with neural functioning, which would then affect the muscles. Two primary conditions that I'll talk about are botulism and myasthenia gravis. And both of them work at the point of the neuromuscular junction, but they work a little bit differently. Now, botulism, if some of you may be taking microbiology, Clostridium botulitum basically is a bacteria that can result in the significant effect of the neuromuscular junction. It results from consumption of contaminated food, typically canned food. Some smoked foods will contain this toxin. The toxin is produced by a bacteria that prevents the release of acetylcholine at the synaptic terminal, and it can lead to a potentially fatal type of paralysis. Now, myasthenia gravis is a progressive muscular paralysis. It affects the loss of the acetylcholine receptors at a junctional fold. The primary cause of this is that our immune system, for whatever reason, begins to attack the acetylcholine receptors and then will basically render those receptors non-functional. It's felt that a genetic factor may play a role which predisposes certain individuals for this condition. And go back to botulism for a second. Now, botulism, and when we talk about the disease process itself, remember there's the bacteria, Clostridium botulitum. The bacteria secretes a toxin. The toxin causes the problem. So the bacteria can be in the stomach. You can treat the bacteria with aggressive antibiotic therapy, but the person may still be symptomatic because the toxins have already been released in the body. So for severe cases of botulism, the person may also have to receive an antitoxin, not just an antibiotic to treat the bacterial infection, to kill the Clostridium that's causing it, but you also would need to be on an antitoxin as well. And I want you to appreciate the concept of how the toxin works. You have a cell, you have a receptor on the cell, muscle cell, where acetylcholine will come and will bind onto the receptor. What the toxin does, the toxin has a stronger affinity or an attraction to the receptor than acetylcholine does. And so what the toxin will do is it'll bind on the receptor, and acetylcholine can't bind on the receptor. And when that occurs, acetylcholine is just left out in that little synaptic space, but it can't work. And so muscles will become paralyzed. The reason why this is so significant is if we paralyze the muscles of the respiratory system, then you can't breathe. So one of the main symptoms that individuals that suffer from botulism, Clostridium botulitum, first you start seeing the GI tract get affected, and then you'll see the respiratory system become affected because it's very sensitive to the ability of the muscles to contract to the diaphragm. So if the diaphragm gets affected, this can lead to a very serious condition. So it's a competitive inhibition by the toxin that causes the problem. So just treating the bacteria may not be enough, especially if enough toxin has been released into the system. Now, in myasthenia gravis, same receptor, acetylcholine binds to the receptor, allows the calcium gates to open up, calcium comes in, causes muscles to contract. Well, in myasthenia gravis, there's no receptor. The immune system, for some reason, begins to recognize the receptors as a foreign invader, and our immune system starts to attack our receptors, and then the receptors no longer can pick up acetylcholine, and the same consequences can occur. Now, there are some temporary conditions that can cause these same types of reactions to take place. There are viral syndromes that can take place that, again, a virus can sometimes mimic our own tissue, and the virus can attach to a receptor, and as our immune system begins to attack the virus kind of changes its coding and begins to look like the receptor. And our own immune system, in an attempt to get rid of the virus, then begins to attack our own system. In this case, it would be a nerve receptor. Now, some of these cases can be temporary, not in myasthenia gravis, but there are some conditions that can be temporary, and a person may have a temporary paralysis for a prolonged period of time, but then as cells start to recycle, new receptors develop, and that the invading virus has been treated or has run its course, then those individuals may recover. But many conditions like myasthenia gravis is a progressive condition, again, an autoimmune process, and so it's not really a medicine we can give that would necessarily replace these receptors. Because it's an immune response, our immune system here will always be attacking the receptors, and so these individuals, typically their life is shortened because of this process. And then another aside that I'll talk about when we talk about muscles is rigor mortis. Those of you who like CSI probably will like this. Because skeletal muscles are involved in our systems, there is a process by which when a person dies that they become stiff. As your book refers to it, stiff as a board. This is a physical state that's referred to as rigor mortis. It lasts until the lysosomal enzymes that are released in the cells will begin to cause autolysis, and then will break down some of the muscle filaments or fibers. And usually that process takes about 15 to 25 hours, depending on the circumstances. Now, environmental factors can affect the breakdown of cells. And remember, lysosomes are in cells. Cells will live so long without oxygen, but at some point, without oxygen, without adequate nutrients, which comes through circulation, and if the body is dead, then eventually the cells will rupture. When they rupture, lysozymes, which are basically proteolytic enzymes, will break out of the cells and then will cause the body to become flaccid. But until that happens, the body typically is in a rigor state. Now, the timing is dependent upon environmental factors. Now remember, when the person initially dies, the body is flaccid. Why? Because recent circulation. And so the body was in its normal process, then the body dies. So there's a period of time where the body would be flaccid. But within usually an hour or 2 hours, you'll start to see some rigidity will develop in the body. And then over the course of the next 15 to 25 hours, the body will be in this state of rigor. Now, if it's in a situation where the room is extremely hot, like in Florida, then this process will be accelerated. And so the lysosomal release will take place a lot faster. If the person is in a very cold environment, the death happens in Wisconsin, the house wasn't heated or whatever, and then the patient may stay in a state of rigor for a much longer period of time. And so when crime labs come in and they're trying to determine the time of death, they look at substances in the body. They may draw some serum and they can measure the lysosomal levels that's in the body, or just from a determination of the state of rigor, it gives them a quick time, that gives them a quick availability to be able to estimate a time of death. You may see, if you watch Law and Order, I like Law and Order, if you watch Law and Order, you may see the coroner, they ask the coroner what time you think the death is. Well, they can tell based on body temperature and the state of rigor whether or not, and there are a series of tests that you do for that, but they can determine almost immediately at the scene at least an approximate time of death based upon this state of rigor mortis. But again, environmental circumstances can play a role in that. So when death occurs, circulation ceases and skeletal muscles are deprived of nutrients and oxygen. Within a few hours, skeletal muscle fibers will run out of ATP at the sarcoplasmic reticulum, and thus calcium ions diffusing into the sarcoplasmic reticulum or the sarcoplasm from the extracellular fluids will trigger a sustained contraction. So without ATP, without the nutrients, again, those cross bridges can't detach from active sites, and so skeletal muscles throughout the body will become locked into this contracted position referred to as rigor until lysosomes are released. Now, some reserves that are important as far as energy goes. Two very important energy packets, ATP, you're already familiar with, and creatine and phosphate. And we'll talk about both of those in just a little bit. Primary function of ATP is to transfer energy from one location to another rather than for long-term storage of energy. At rest, skeletal muscle fibers produce ATP. They typically will produce more than it actually needs, and then under certain conditions we'll see the transfer of energy to another energy package referred to as creatine. Creatine is a small molecule that muscle cells will assemble from fragments of amino acids. The energy transfer creates a high energy compound referred to as creatine phosphate, which we will see in skeletal muscles and some smooth muscle. The enzyme that facilitates the release of this reaction is referred to as creatine phosphokinase, CPK. When muscle cells are damaged, if you're looking for muscle injury, CPK, which again is the enzyme, gets released from cell membranes, and it gets released into the bloodstream, and it allows us to be able to measure where the muscle damage has taken place. Now, high blood concentrations of creatine phosphokinase, or CPK, usually indicate serious muscle damage. Now, CPK is in almost every skeletal muscle and every smooth muscle and every cardiac muscle. And so when muscle gets damaged, we see the release of CPK. Now this is where CPK is important. If a person has a heart attack, comes into the emergency room with chest pain, one of the first labs that we do is we obtain a CPK, creatine phosphokinase, because we want to see if the heart muscle has been damaged from the heart attack. Okay, CPK is measured and the CPK is elevated. But if the person was involved in a car accident, or if you even just punched them in the arm and they got a big bruise on their arm and you bruised a muscle, that would also elevate CPK. So CPK is indicative of muscle damage, but it's not really specific until we look at CPK further. Now each CPK is specific for each different muscle. CPK in the skeletal muscle has a particular constituent. CPK from the cardiac muscle has specific constituents. CPK from smooth muscles of the gastrointestinal tract have different markers as well. So each of these particular muscles, the CPK there has particular markers. So after we see that the CPK is elevated and we know that there has been muscle damage, then we fractionate that even further to find out if the CPK is from cardiac, if it's from skeletal muscle, if it's from smooth muscle of the GI tract. But it's a very important marker in diagnosing a person that comes in with a heart attack. So CPK, creatinine phosphokinase, is a very important marker. High blood serum indicates that the person has damage to a muscle, but again, you have to be a little more specific to be able to find out whether that muscle is from the heart, whether it's from skeletal muscle or wherever. So creatinine phosphokinase, very important in clinical diagnosis, and you'll see that again in a clinical setting where persons had their CPK drawn to identify whether or not they've had a heart attack or not. Another concept that you need to appreciate in muscles is the process of aerobic metabolism. Now there are two processes. One is aerobic metabolism in the presence of oxygen. Another one is anaerobic metabolism in the absence of oxygen. Cells in the body generate ATP through a series of steps. Aerobic metabolism occurs in the mitochondrium. Remember when we talked about the Krebs cycle taking place, glycolysis taking place outside the mitochondria in the cytoplasm. The 3-carbon sugar brought into the mitochondria goes through these series of enzymatic steps referred to as the Krebs cycle, TCA cycle, citric acid cycle. Takes place, and we see the generation of ATP in the presence of oxygen, and we get a lot of ATP from that process. Aerobic metabolism provides 95% of ATP demands for resting cells. Mitochondria will also absorb oxygen, ADP, phosphate ions, organic substrates from the surrounding cytoplasm. Now, because it's aerobic, it provides us a significant amount of energy. Thus, when we talk about aerobic endurance, we're talking about the ability of muscles to sustain prolonged activity because they are supported by mitochondria, they are supported by nutrients and oxygen that's available there in the cell. Determined primarily by the availability of substrates from aerobic respiration, which muscle fibers can obtain by the breakdown of carbohydrates, lipids, and or other amino acids. Initially, many of the nutrients get catabolized by muscle fibers or obtained from reserves in the sarcoplasm. Prolonged aerobic activity must be supported by an adequate amount of nutrients circulating in the blood, as well as an adequate amount of oxygen that must be available and circulating in the blood as well. Aerobic metabolism again aerobic metabolism takes place inside the mitochondria. Now, when we talk about anaerobic metabolism, in the absence of oxygen, this process takes place in the cytoplasm. The process that we've talked about previously referred to as glycolysis. This is the breakdown of glucose. Remember, glucose gets into the cell by using insulin. Insulin helps facilitate, there are insulin receptors outside the cell that actually will bring glucose into the cell. Glucose, 6-carbon sugar, brought into the cell, but for the 6-carbon sugar to get inside the cell, it's got to be broken down into a 3-carbon structure. That takes place in the cytoplasm, referred to as glycolysis. Breakdown of glucose into pyruvic acid, which is a 3-carbon compound, a 3-carbon molecule, in the cytoplasm. This is an anaerobic process, doesn't require oxygen. Glycolysis provides a net gain of about 2 ATP from molecules and will generate 2 pyruvic acid molecules from the glucose, and these 2 pyruvic acid molecules will then pass through the membrane of the mitochondria and then will go through the TCA cycle. ATP produced by glycolysis is therefore only a small fraction of that produced by aerobic metabolism, which again breaks down the 2 pyruvic acids in the mitochondria, which would generate 34 ATP molecules. Now, anaerobic process of glycolysis enables cells to generate additional ATP when mitochondria are unable to meet current needs. Now, even though glycolysis doesn't produce much ATP, and even though it's an anaerobic process, sometimes it's just that little amount of ATP, that little amount of energy that's produced in this anaerobic process that can sustain the body for a short period of time. Now, anaerobic endurance is not long sustained endurance like we see in aerobic metabolism. In aerobic metabolism, the mitochondria are producing plenty of energy, they're getting plenty of nutrients, plenty of oxygen. Anaerobic endurance basically can't be supported for long periods of time. Length of time of muscle contractions can continue to be supported by glycolysis, by existing energy reserves of ATP and creatine and phosphate. Anaerobic endurance, however, is very limited. And it's limited because of the amount of ATP and creatine and phosphate that's typically on hand, the amount of glycogen available. Now, glycogen is the storage unit for glucose, typically in the liver and in the muscle. Glycogen is stored there, can be broken down and release more sugar. But in that process of when glycogen is being broken down, acid is produced, lactic acid is produced, and lactic acid changes the body's pH. Normal body pH, 7.35 to 7.45, as we talked about earlier. If a lot of lactic acid is produced, it changes the acidity of the body. And so in sustained, in an attempt to maintain sustained anaerobic endurance, when more of this lactic acid is being broken down, it changes the acidity of the body, and as we all know, that's not good. So typically the onset of muscle fatigue will occur within 2 minutes of the start of maximal activity. Now, we need anaerobic metabolism. Don't misunderstand what I'm saying. Not very efficient, but we need it. Why do we need aerobic metabolism? Well, we need pyruvic acid, number one. Pyruvic acid, remember, goes into the cell and will eventually, through aerobic metabolism, will produce more energy. So that's a good thing. But the anaerobic metabolism as well, an example, let's say the heart stops beating. Heart stops beating, you don't circulate oxygen, you don't circulate nutrients, and so the heart stops beating. What happens? Well, there's no aerobic metabolism, so until we can start CPR and get circulating started and do all the fancy stuff that we do in hospitals with shocking the heart and giving different types of cardiac medicines, we need some anaerobic metabolism to sustain the body, even though it's just for a short period of time. But it may give us that 2 to 5 minutes that we need to sustain body function until we can get the system working again. So anaerobic metabolism is important. Even though it doesn't produce a lot of energy, it's important in being able to sustain the body for short periods of time, and then certainly the anaerobic processes of generating pyruvic acid are important to the body as well. Now, the concept of lactic acid, again, when the body is running out of an energy source, running out of nutrients, glucose, then the body sends a signal to the liver to release the stored glucose, the glucose stores, which is stored in the form of glycogen. This process is referred to as glycolysis, where glycogen is broken down to produce more sugar. Now, when this happens, it also produces lactic acid. So as glycolysis produces pyruvic acid faster than utilized by the mitochondria, pyruvic acid levels will rise, and under these conditions, pyruvic acid would be then converted to lactic acid, which again is related, but lactic acid is very acidic, less than 7.35. The problem with that is lactic acid, as it changes the pH, significantly affects body functioning. Lactic acid diffuses out of the muscle fibers into the bloodstream. Process continues until the exertion is ended because intracellular lactic acid concentrations will be relatively high. Liver will absorb the lactic acid and convert it to pyruvic acid, which is an important part of the process because we need pyruvic acid. But 30% of the pyruvic acid molecules are broken down in a TCA cycle, providing ATP for energy. But again, the downside of lactic acid is, because it's an organic acid, dissociates in the body fluid, increasing the hydrogen ion concentration, therefore affecting intracellular pH, dropping intracellular pH below the level of 7.35. Below the level of 7.35, our body doesn't work well. The enzymes that carry out cellular function in our body don't work well in a pH less than 7.35, and thus the production of a significant amount of lactic acid for energy can cause significant problems in the body. Now, there are some buffers in the muscular system that will resist the pH shift for a period of time, but it's very limited. Eventually, the changes in the pH will alter the functional characteristics of key enzymes, and the muscles will fail to function. So, big differences between aerobic and anaerobic metabolism. As you may be familiar with, some individuals, athletes, use interval training, a combination of aerobic and anaerobic metabolism, to induce a better level of performance. Aerobic activities do not promote muscle hypertrophy. Anaerobic metabolism will produce muscle hypertrophy, and so because of that, we see a combination of aerobic and anaerobic exercise. Now, hypertrophy, which is a term that we'll talk about in just a little bit, it's a process by which muscle fibers get bigger. Now, muscle fibers don't increase in number, they actually get bigger. And important to recognize that. So a lot of athletes do anaerobic training along with aerobic training to make their muscles get bigger. And this is referred to as interval training. Interval training particularly useful in persons engaged in racket sports, tennis, squash, dominated by aerobic activities, punctuated with periods of anaerobic effort. You see that weightlifting, for example. Weightlifting is an anaerobic process. Now, interval training using exercises that increase heart rate, increase respiration, and then jumping into doing some weightlifting is a form of interval training where you're using anaerobic metabolism and aerobic metabolism, and it actually will facilitate the growth and development of muscles. Now, what happens when muscles become hypertrophied or get bigger? Basically, mitochondria and other nutrients that the cell requires. When you exercise muscles by lifting weights, the muscles have memory, and they recognize, well, I need a tremendous amount of energy source built up already because I'm not going to get it at the time when the person's doing the weightlifting. So mitochondria and other protein elements and nutrients will pack into those muscle fibers, and it takes space, and so the muscles will begin to get bigger. Now, it doesn't increase the number of fibers, it's just the fibers get bigger, and that's how hypertrophy takes place. In many exercises or exercise activities, we see muscle soreness, and many of you who've worked out before may experience muscle soreness, periods of physical exertion. Considerable controversy on what actually causes that pain. There are some debates whether or not when a person does training, aerobic or anaerobic training, whether or not small tears in muscle tissue occurs and little enzymes are released into the system. That can cause pain, stimulate the pain receptors. Whether or not it's from muscle spasms, the muscles, because acetylcholine has been released, acetylcholinesterase has been released so repetitively, the muscles now go into kind of a state of tetany or spasms. That can lead to the muscle pain. Tears in the connective tissue, not the muscle itself, but the connective tissue fibers, whether or not that area was damaged or not. So it's still a lot of debate about what causes muscle soreness. Some argue that lactic acid being spilled in these areas can also cause pain as well. So whatever your train of thought is, that's just a little aside. Another process inside the muscle, glucose molecules are basically released into circulation and absorbed by skeletal muscles and used to rebuild glycogen reserves. This is called the Cori cycle. It's the shuffling of lactic acid to the liver and glucose back to the muscles. That's basically called the Cori cycle. Again, lactic acid to the liver, glucose back to the muscle cells are involved in the process of being able to generate the nutrients that's required by the muscle. Again, that's referred to as the Cori cycle. I'll let you guys take like a 5-minute break, and then we'll come back and we'll talk about hormone and muscle metabolism, and we'll get into one of the small groupings of muscles so you have some anatomical structures to study and look at. Okay?